Okay, so welcome back to this next video in which we are discussing the mammalian circadian timing system. Okay, so we're currently in the process of discussing the glutamate receptors so that we can understand how they're going to generate a signal which is going to be a calcium signal within the suprachiasmatic nuclei neurons, and how this is going to help re-entrain our suprachiasmatic nuclei neuron to the light-dark cycle, or keep it in tune with the light-dark cycle. Okay, so we've just done the Nmda receptors. We're now going to turn our attention to the kainate receptors, which is another family of ionotropic glutamate receptors. Okay, right. So the kainate receptors are often abbreviated to the KARs for short, for kainic acid receptors, and kainic acid is just a agonist for the kainic acid receptors. Okay, so it will bind to the ligand binding domains just like glutamate molecules would and trigger the kainate receptors to open. Okay, right. So there are 5 subunits for kainate receptors, okay? So these are called gluK1, gluK2. These are their new names. Again, I'll tell you what their old names are because, again, in older papers you will see their old names used. Then there is gluK3, gluK4, and gluK5. And again, all of these subunits have that same basic structure that we've discussed now, okay? They all have this same basic structure here, and they will tetramerize into tetramers to make the full ionotropic glutamate receptor, or the full kainate receptor in this case. So we now want to discuss, just like we did with the Nmda receptors, how can the kainate receptors subunits join together to form these tetramers, which are the full kainate receptors. Okay, so let's start off with the known homotetramers. Okay, so I didn't tell you the old names for them. Let me firstly just tell you the old names for them. So, remember when we were discussing the old names for the Nmda receptor subunits, we saw that the GluA1 to GluA4 subunits were called GluR1 to GluR4 in the old naming system. Well, we continue the GluR naming system on. So GluK1 used to be called GluR5, GluK2 used to be called GluR6, and GluK3 used to be called GluR7. Finally, GluK4 and GluK5 used to be called kainic acid 1 and kainic acid 2. Okay, so those are the old names, and certainly if you read older papers and older reviews, you will see these old names used. Right, okay, so let's now discuss how these subunits can join together to form full kainate receptors. Okay, so firstly let's discuss the homotetramers. So, gluK1 through gluK3 are all capable of forming homotetramers, which means that you can take 4, for instance, gluK1 subunits, join them all together, and that will form you a gluK1 homotetramer. You can do the same thing for gluK2 and the same thing for gluK3. So these are 3 homotetrameric kainate receptors which you can form. Okay, now to discuss the heterotetramers which you can form. Again, the heterotetramers which we know actually exist are all diheterotetramers, just like was the case in the Nmda receptors. Okay, so you can't go too wild. You can only build tetramers which have 2 different types of kainate receptor subunit within them. Okay, now what are the rules then for building a diheterotetramer? So you have to firstly decide which 2 types of kainate receptor subunits you're going to use, and these are the rules for how you can pair them together. Okay, so basically you need to pick 1 kainate receptor subunit from the ones that are circled in red. So you pick 1 of gluK1, gluK2, gluK3. So pick 1 of these, and then pick 1 of these ones that are circled in orange, gluK4 or gluK5, and then that is a diheterotetramer you can form. For instance, I might pick gluK2 here, okay, out of the red, and now I need to pick 1 from the orange, so let's pick gluK4, and that is a diheterotetramer which you can find. Okay, so you can find kainate receptors which contain both glucate-2 subunits and glucate-4 subunits. Okay, so in total that gives us 6 possible diheterotetramers. You can find glucate-1, glucate-4 diheterotetramers, glucate-1, glucate-5 diheterotetramers, glucate-2, glucate-4 diheterotetramers, glucate-2, glucate-5 diheterotetramers, glucate-3, glucate-4 diheterotetramers, and then finally glucate-3 paired with glucate-5. Okay, so those are the diheterotetramers you can find. And those now are the kainate receptors. Now, what we also want to discuss is what the effect of opening a kainate receptor via binding glutamate to it is going to be. Well, this is almost identical to the AMPA receptors. Okay, so basically the kainate receptors are permeable to both sodium and potassium ions, but not to calcium ions, so they don't have permeability to calcium at all. Okay, but it's the exact same setup as for the AMPA receptors. When you open this channel for sodium and potassium, you're going to get a net movement of sodium in, okay, down its concentration gradient and the electrical gradient, and you're going to get a slight movement of potassium out, because the concentration gradient favours the movement out, whilst the electrical gradient opposes that and says move in, okay, and when you subtract one from the other, you get a slight net movement of potassium out. Okay, this overall means you're going to move more positive charge into the cell than out, and this is going to form your depolarizing current, which will depolarize the electrical potential difference across the cell membrane, causing an excitatory postsynaptic potential. Okay, so the kainate receptors are very similar to the AMPA receptors. They're made up of different subunits than the AMPA receptors, but effectively when you open them they do the same thing to the cell. Okay, so on these suprachiasmatic nuclei neurons then, you're going to have AMPA and kainate receptors, and the glutamate that is released by the axon terminal of the photoreceptive retinal ganglion cell is then going to act on these AMPA and kainate receptors and cause them to open, and that will trigger depolarization of the electrical potential difference across the cell membrane of these suprachiasmatic nuclei neurons. Okay, and that's going to contribute to the opening of the Nmda receptors, as we'll see. Okay, so Nmda receptors not only require glutamate to open, but they also require a depolarization, and the depolarization is going to be produced by the kainate and the AMPA receptors. Okay, so we're now going to turn our attention to the Nmda receptors, which are either the most interesting or the most difficult, depending on how you see it. Okay, right, so Nmda receptors then. Okay, so again, Nmda receptors all have that same basic structure that all ionotropic glutamate receptors share. They're all tetramers, and they all of the subunits of Nmda receptors have that same basic structure. Okay, so we now want to discuss the different types of Nmda receptor subunit. Okay, so there are 7 different Nmda receptor subunits, and then we'll discuss how you can assemble these 7 Nmda receptor subunits together to make full Nmda receptors. So the first Nmda receptor subunit is GluN1. You then have 4 GluN2 subunits. Okay, you have GluN2A, GluN2B, GluN2C, and GluN2D, and you'll notice that all of these subunits begin GluN. Okay, and the N obviously stands for Nmda, the A in GluA stood for AMPA, and the K in GluK stood for kainate. Okay, so here we have GluN2C, and then finally GluN2D. Okay, so those are all separate Nmda receptor subunits, but they're all classified as the GluN2 family of Nmda receptor subunits. Then finally, there is also the GluN3s, and in this there are GluN3A and GluN3B. Okay, right, so we now want to discuss how from these 7 different Nmda receptor subunits you can build a whole Nmda receptor. Okay, right, so the first thing that I need to tell you is that absolutely always in an Nmda receptor you have 2 GluN1 subunits. Okay, so let me show you this. So let's say we are now looking at our NMDA receptor from above. So we're looking from the extracellular aspect down at the 4 quarters of the NMDA receptor, which are these 4 separate proteins, okay? And here is the pore down the middle. So, absolutely always, 2 of these subunits of NMDA receptors will be GluN1 subunits. Okay, and I'm highlighting these in blue here. So these 2 are both going to be GluN1 subunits. Okay, so in absolutely every single NMDA receptor you ever build, you will have 2 GluN1 subunits like so. Okay, now, the only question that then remains is what are these other 2 subunits? Okay, and that's got a big answer, basically. There are many different subunits that can be. The only thing I can say that they're definitely not is they're not GluN1 subunits. They're going to be either GluN2 subunits or GluN3 subunits, or they can even be a mixture of the 2. Okay, so we'll start off then with the GluN1, GluN2 diheterotetramers. Okay, so GluN1, GluN2, and this time they're going to be diheterotetramers. So remember what diheterotetramer means. It means that the tetramer is going to have 2 different subunits within it. Okay, 2 of them are already GluN1, so we've only got 1 subunit left to play with. Okay, and it's going to be a GluN2 subunit. So what these GluN1, GluN2 diheterotetramers are is they have 2 GluN1 subunits here, and then they have 2 GluN2 subunits here, and these 2 GluN2 subunits need to be the same type of GluN2 subunit. So for instance, if this is a GluN2A subunit, then this one also has to be a GluN2A subunit. Otherwise it wouldn't be a diheterotetramer. It wouldn't only contain 2 different types of subunits. Okay, so you can make many different GluN1, GluN2 diheterotetramers. You can make it where there are 2 GluN2As here, where there are 2 GluN2Bs here, where there are 2 GluN2Cs here, and where there are 2 GluN2Ds here. So that's 4 NMDA receptors that we've now discussed that are under this category here. Okay, so let's move on to the next category, which are the NMDA receptors where you still have 2 GluN2 subunits here, but they don't have to be the same. Okay, so these are going to be triheterotetramers. Okay, so triheterotetramers. So it's the first time we've seen triheterotetramers, where at this time you're going to have 3 different subunits. Okay, so let me give you an example of one of these triheterotetramers. Okay, and there are 4 examples that are well known. Okay, so I'll give you those 4 examples. So I'll draw one of them out for you. So you'll have, as always, your 2 GluN1 subunits here. Okay, and we'll colour those in blue here. So here is 1 GluN1 subunit, and here is our second GluN1 subunit here. But now you have 2 GluN2 subunits here, but they're not necessarily the same. So this one, for instance, could be GluN2A, and this one here could be GluN2B. Okay, so let's highlight GluN2A in orange here, whoops, like so, and then we'll have GluN2B in red here. Okay, so what other GluN1, GluN2 triheterotetramers can we form? Well, here's one where we've paired GluN2A with GluN2B, but you can also pair GluN2A with GluN2C and also with GluN2D. So just swap the B here for a C or the B for a D, and those are also GluN1, GluN2 triheterotetramers. Okay, finally, another example is you can also have the GluN1, okay, GluN2A, sorry, not GluN2A this time, GluN2B, GluN2D triheterotetramer. That's another example in which you will have 2 GluN1 subunits as always, 1 GluN2B subunit here, and then 1 GluN2D subunit as well. Okay, right, so those are triheterotetramers, which again still consist of 2 GluN1 subunits and 2 GluN2 subunits. So we haven't seen an involvement yet of GluN3 subunits. Okay, so now that's about to change. Now let's talk about gluN1, gluN2, gluN3 triheterotetramers. Okay, so these are Nmda receptors which have two gluN1 subunits, as always. Okay, then they'll have one gluN2 subunit and also one gluN3 subunit. Okay, so let's draw a picture of this. So again, if we're viewing from the top of the receptor, then we've got the pore down the middle here, like so. Okay, we've split it up into these four separate subunits. Here we have two gluN1 subunits, and then we'll have one gluN2 subunit, and then also one gluN3 subunit. Okay, so in blue here, these are both gluN1 subunits, one there, and another one here. Okay, then we've got a gluN2 subunit here in turquoise, and that can be one of the four. Okay, and then we've also got a gluN3 subunit here in green. Okay, so those are gluN1, gluN2, gluN3 triheterotetramers. Finally, you can also have subunits which Nmda receptors which don't contain any gluN2 subunits. Okay, and these will be called gluN1, gluN3 tetramers. Okay, and you can have diheterotetramers and triheterotetramers. So let me explain what I mean by this. So we can have a N1, gluN3 diheterotetramer in which we only have two different types of Nmda receptor, which means that we can only then have one type of gluN3 subunit making up the other two slots. So if I draw a picture of this here, here is our Nmda receptor view from above. We have two gluN1 subunits we know here, and then we'll have two gluN3 subunits here, but this time both of these gluN3 subunits will be the same type, so they'll either be gluN3A, both of them, or they'll be gluN3B. Okay, so let's colour that in. So here we have our two gluN1 subunits in blue here. Okay, and then these two are at the moment identical, and that will give you a diheterotetramer because we've only got two different types of subunit making up the entire tetramer. Okay, you can also make triheterotetramers. Well, you can make one type of triheterotetramer, which is a gluN1, gluN3 triheterotetramer, and this is going to contain two gluN1 subunits, as always, one gluN3A subunit, and then also one gluN3B subunit. Okay, like so. So one of these would be an N3A, and then the other would be an N3B. Okay, so that's the different types then of Nmda receptors which you can form. Okay, right. We now need to discuss the ligands for these Nmda receptor subunits, because it's not as simple as it was for the AMPA and the kainate receptor subunits. The AMPA and the kainate receptor subunits, their ligand binding domains all bound glutamate. Okay, it's not the case for the Nmda receptor subunits. In fact, only the gluN2s actually bind glutamate at all. Okay, so these ones in the center here that I'm now highlighting in green, they have as their ligand glutamate. Okay, so glutamate activates these. But the other three, the gluN1 subunits, which we always have within our Nmda receptor, and the gluN3 subunits, okay, so I'll circle these in red, so these ones and these ones here, they have a different ligand, basically. They have a ligand requirement that is either glycine or D-serine. Okay, now usually in a physiological circumstance their ligand will be glycine rather than D-serine, because D-serine is quite rare. It's quite rare to have D forms of amino acids. So let me just discuss the structure then of glycine and D-serine, which are the ligands for these gluN1 and gluN3 Nmda receptor subunits. Okay, so those do not bind glutamate. The gluN2s bind glutamate, but the gluN1s and the gluN1 and the gluN3A and gluN3B subunits, they don't bind glutamate. They need glycine or D-serine instead. So let's start with glycine, which is nice and easy, and then we'll discuss D-serine, which is more difficult because I'll have to discuss optical isomerism with you. Okay, right. So So let's start with the core structure of an amino acid. Here's the amino group, here's the alpha carbon with a hydrogen coming off it, and here's the carboxylic acid group down here. Okay, and the R group of glycine consists of a single hydrogen atom attached to that alpha carbon. Okay, so this is the structure then of glycine. Okay, and I'll circle this in blue. Now let's talk about the structure of that other ligand, which is D-serine. Now I want to stress that in reality the usual ligand will be glycine, not D-serine. I'll stress that again. But D-serine is also a ligand. So, let's start off with a discussion of the difference between an L and a D enantiomer of an amino acid. Okay, so basically, usually with amino acids, where the R group is more dramatic than a single hydrogen, okay, usually this carbon here, the alpha carbon, is a chiral center. It's not a chiral center in the case of glycine, which is why you don't need to worry about whether this is L or D glycine. There's no such thing. There's only one type of glycine because this carbon isn't a chiral center. In order to be a chiral center, you need to have 4 different groups coming off you. This carbon has 2 identical groups here in the form of these 2 hydrogens, and that means that it doesn't actually have 2 different optical isomers. Okay, but more general amino acids, where you have a more complicated R group here, are going to be, optically active molecules, as they're called, molecules which have different optical isomers. Okay, and let me explain why. And to explain this, I need to show you the 3-dimensional structure of these amino acids. Okay, and this is difficult because I have to draw a 3-dimensional structure. Okay, so let's put the alpha carbon right at the center here. Okay, and at the moment we're just working with an arbitrary amino acid because most amino acids do have a D enantiomer and an L enantiomer. Okay, so I'll discuss this in general for all of the amino acids and then we'll apply it to the amino acid serine. Okay, so let's say we've got the alpha carbon positioned nicely in the plane of my piece of paper. Okay, so that alpha carbon sitting on my piece of paper. Okay, then it's bound to a hydrogen always. This is the hydrogen that comes off the alpha carbon in all amino acids. Okay, and we'll say that that's also in the plane of my piece of paper. I can do that. I can orient the molecules so that the carbon and the hydrogen are in the plane of the piece of paper. Okay, let's also say that I'm going to have my amino group here, also positioned in the plane of the piece of paper, and this is the end of how far I can go with trying to get my groups positioned in the piece of paper. Once I've got 3 like this, I'm stuck with where the others end up. Okay, so these are all sitting nicely in the plane of the piece of paper. The other 2 groups, the carboxylic acid group and the R group, one is going to have to come out of the page towards us in this sort of a direction. The other is going to have to go into the page away from us. Okay, now the question is, which one goes into the page away from us and which one comes out of the page towards us? Okay, so just to give an example, let's say that the carboxylic acid group is coming out of the page towards us, and to show that a group is coming out of the page towards us, you use one of these bonds where it gets thicker towards the end, basically. Okay, and here is the carboxylic acid group. Okay, so the carboxylic acid group is coming out of the page towards us, whilst the R group is going to go into the page away from us. And to show going into the page away from us, I'll put a dashed line like so. Okay, so this R group is going to go into the page away from us. Okay, so what if I did it the other way around? Well, those 2 molecules now would not be the same thing. Okay, so if I draw the other one now, okay, so here is the carbon, here's the hydrogen, here's the amino group. They're all still in exactly the same orientation, but now I'm going to swap around which one of these groups comes out of the piece of paper. I'm going to say the R group is going to come out of the piece of paper, and the carboxylic acid group is going to go into the piece of paper. Okay, this is not the same as this. There is no way that you can just rotate this around, or rotate yourself, better yet, change the way you look at this. There's no way by changing the way you look at this you can turn this into this, okay, because you have to stay looking at it in the same way so that the alpha carbon, the amino group and the hydrogen are superimposed on these ones, okay. Without breaking this bond and breaking this bond and swapping the 2 groups over, you can't turn this into this. So you can't turn this into this without performing a chemical reaction, basically. Okay, so they are different molecules. They are the two optical isomers of the amino acid. Now, in this case, these optical isomers are actually very special optical isomers of one another. Okay, this isn't true for more complicated optical isomers necessarily, but in this case it's beautiful, because this one is actually the mirror image of this one. So if you imagine what would happen if this one was to look in the mirror, obviously it would not see this, but if you rotated this around by 180 degrees, that would be the mirror image of this. So imagine rotating this around by 180 degrees. That will bring this R group over to this position, so it will be opposite this one. That will bring the carboxylic acid group coming out of the page over here, so they'll be opposite this one, and the amino group will end up going away into the page away from us. Okay, so is the mirror image of this one. Now, when two optical isomers of a molecule are mirror images in this way, there's a special name for that. They're known as enantiomers. Okay, that's the spelling of it. Okay, so they're known as enantiomers. So this is one enantiomer of the amino acid, this is the other enantiomer of the amino acid. Now there is a special naming system for the enantiomers of amino acids. Okay, one's going to be called the L enantiomer, and the other's going to be called the D enantiomer. Okay, now, this one here is the L enantiomer, and I'll tell you how I know that in a moment. Okay, so this is the L enantiomer, and this one here is the D enantiomer. Okay, now what does L and D stand for? Well, L stands for larvo-rotatory, okay, and it's all to do with how it rotates light or something along those lines. And then D stands for dextro-rotatory. Okay, right. Basically, how do I know which one's the L enantiomer and which one's the D enantiomer? Well, there is a nice trick that you can use, and it is, imagine sitting where the hydrogen is sitting. So imagine sitting up here with this hydrogen that comes off the alpha carbon. So imagine that you are sitting on top of this alpha carbon's hydrogen up here, and you are going to try and read the word corn. Okay, now how do you read the word corn? Well, you start with the CO, which comes from the carboxylic acid group. You then move on to the R, which is the R group, and then finally the nitrogen, which is the nitrogen of the amino group. Okay, so you're going to try and read the word corn. Okay, so which direction will you have to turn your head? Well, you'll start down here looking at the CO of the carboxylic acid group. You'll then turn your head to the R over here of the R group, and you'll then turn your head to the nitrogen for the final bit of corn. So your head will go in this direction. It will turn clockwise, basically. You'll have to rotate your head clockwise. Okay, so if you have to rotate your head to read the word corn, that's the larvo-rotatory enantiomer. So this is the clockwise one. Now let's have a look at the same thing if we are sitting on top of a D enantiomer here. Okay, let's try and read the word corn again. So we'll start with the CO from the carboxylic acid group here. We'll then turn our attention to the R, and then finally to the N. Now, which direction will we rotate our head this time? Well, we'll be rotating it in the anti-clockwise direction, like so. Okay, and when you have to rotate your head in the anti-clockwise direction to read the word corn, that's the dextro-rotatory enantiomer. Okay, so that's the difference between D and L enantiomers of amino acids. Okay, so most amino acids within cells are in the L enantiomer form. Okay, so it's quite rare to find an amino acid that's in this D form. However, these GluN1, GluN3A, GluN3B subunits are specifically activated by the D form of the amino acid serine. So let me just show you the R group of serine. In fact, I'll put it here. The R group of serine is then just a methylene group with an alcohol group. Okay, so imagine substituting this in for this R group. That certainly makes this carbon a chiral center, because now it's got 4 different groups coming off it. Okay, and if you put it in on this structure here, you'll get D-serine then. Okay, and this is the ligand for GluN1 subunits, GluN3A subunits, and GluN3B subunits, but the more likely ligand is glycine. Okay, right. So this has a number of consequences because now only the GluN2 subunits are actually sensitive to glutamate. Okay, the GluN1 subunits are going to need either glycine or D-serine bound to them, as are the GluN3A or GluN3B subunits. So let's look through our Nmda receptors and try and understand what this means. Okay, well, if we start with these GluN1, GluN2 diheterotetramers or triheterotetramers, so these are the Nmda receptors where you have two GluN1 subunits and then two GluN2 subunits that are either the same, making a diheterotetramer, or are different, making a triheterotetramer. Okay, you are going to need to open your GluN1, GluN2 Nmda receptor. You're going to need two glycine or D-serine molecules, one to bind to each of these GluN1 subunits, and you're then going to need two glutamate molecules to bind to your two GluN2 subunits. Okay, now, in the case of these GluN1 subunits, it's not too much of a problem because generally the concentration of glycine in the extracellular fluid is large enough that the GluN1 subunits permanently have glycine bound to them. Okay, so let's draw this glycine here and this glycine here and a glycine here and a glycine here. So usually the GluN1 subunits have glycine permanently bound to them at the extracellular fluid concentration of glycine. That means that to open these GluN1, GluN2 Nmda receptors, all you need is two glutamate molecules to come and bind, one to each of the two remaining Nmda receptor subunits, the two GluN2 subunits. Okay, and this will then cause the Nmda receptor to open. Okay, right, so the fact that the GluN1 subunits need glycine isn't too much of a problem, basically. It means overall that usually all you need is glutamate because the GluN1 subunits usually have the glycine already bound to them, basically. So all you need is the glutamate, and effectively these are now glutamate gated. Now let's work our way down. Going to the GluN1, GluN2, GluN3 triheterotetramers, which are Nmda receptors where you have two GluN1 subunits and then a GluN2 subunit and a GluN3 subunit. This time, both the GluN1 subunits and the GluN3 subunit that you have here, all three of those are going to require glycine bound to them, or D-serine, but we'll forget about D-serine because that's not going to be present in any decent amount, so we'll have glycine, okay? So we need a glycine bound here, and a glycine bound here, and a glycine bound here, and then a glutamate bound here. Now, these are slightly less likely than the GluN1, GluN2 subunits to have three glycines bound to all three of these subunits. Okay, so to an extent, these ones may well be affected by glycine concentration as to whether they activate. So they're certainly more sensitive to glycine than the GluN1, GluN2 Nmda receptors. Okay, so potentially these ones may well require some glycine and some glutamate to open, and might not just have all three sites occupied with glycine prior to an increase in the glycine concentration. Okay, so those ones are a little bit more interesting. Then GluN1, GluN3 subunits. These are the ones, GluN1, GluN3 Nmda receptors. These are the ones which consist of two GluN1 subunits and then two GluN3 subunits. And they're either diheterotetramers, which means that both of these GluN3 subunits are the same type, or they're triheterotetramers, which means you have one GluN3A and one GluN3B. Okay? All of these subunits require glycine or D-serine, not glutamate. So these are actually not glutamate receptors at all. Okay, they're still called glutamate receptors because they have the same structure as the ionotropic glutamate receptors, but their ligand is actually completely glycine. Okay, and this was a big surprise when people first realized it, because glycine is a neurotransmitter used within the spinal cord mainly. Okay, and there it acts on special receptors called glycine receptors, which are not like this at all. They are ligand-gated ion channels, but they're cis-loop ligand-gated ion channels, which contain 5 subunits rather than 4, and also have a completely different subunit structure. Okay. Furthermore, the glycine receptors in the spinal cord open and are a chloride channel. okay, and allow chloride anions into the cell, hyperpolarizing the cell membrane, so they reduce the excitability of the postsynaptic neuron. Okay, so they're inhibitory receptors, basically, whereas these are excitatory receptors, and it wasn't known that there were going to be excitatory glycine receptors within the brain. Okay, so we're not going to be too interested in these ones. We're going to be interested in the ones which actually are receptors for glutamate, which are the GluN1, GluN2 Nmda receptors, and also these GluN1, GluN2, GluN3 subunits. That's fair. GluN1, GluN2, GluN3 Nmda receptors, rather. Okay, so it's fair to say that these ones are the main ones that are found in the brain. You have the GluN1 with GluN2 subunits, okay? The ones that are actually gated solely really by glutamate, because they always have the glycine bound to them. Okay, so these are the ones we'll focus on. Okay, so in the next video what we'll do is we'll discuss when glutamate binds to these two GluN2 subunits of the Nmda receptor, which is either a GluN1, GluN2, well, which is a GluN1, GluN2 Nmda receptor. Okay, how is that actually going to affect the electrical potential difference across the cell, and moreover, how is that going to create us a calcium signal? Okay, so we'll look at how these Nmda receptors open